Hi class, this is your instructor, Skylar Huff. This is Chapter 23, Systematics, Phylogenies, and Comparative Biology. All organisms share many biological characteristics. They are composed of one or more cells, carry out metabolism, and transfer energy with ATP, and encode hereditary information in DNA. Yet there is also a tremendous diversity of life ranging from bacteria and amoebas to blue whales and sequoia trees. The picture you see here, what it does is it shows a variation in one particularly diverse group of organisms. That diverse group of organisms is known as a beetle, or beetles in general, from order Coleoptera. Beetles are the most diverse group of animals. Of the approximate, let's say, 60% of all animal species are insects, there are far more species of beetles than any other type of insect. And then among the beetles themselves, families that are herbivorous are particularly species rich. So for generations, biologists have tried to group organisms based on shared characteristics. The most meaningful groupings are based on the study of relationships among organisms. New methods for constructing evolutionary trees and a sea of molecular sequence data are leading to improved evolutionary hypotheses to explain life's diversification. So let's get into the chapter. One of the great challenges of modern sciences is to understand the history of ancestor-descendant relationships that unite all forms of life on Earth. And that was going to be from the earliest single-celled organisms to the complex organisms we see around us each and every day. If the fossil record were perfect, we could trace the evolutionary history of species and examine how each arose and proliferated. However, as discussed in Chapter 21, the fossil record is far from complete, answering many questions about life's diversification, but leaving many others unsettled. Consequently, scientists must rely on other types of evidence to establish the best hypothesis of evolutionary relationships. Bear in mind that the outcomes of such studies are hypotheses, and such they require further testing. All hypotheses may be disproved by new data, leading to the formation of better, more accurate scientific ideas. The study of evolutionary relationships is called systematics. By looking at the similarities and differences between species, systematists can construct an evolutionary tree or phylogeny, which represents a hypothesis about patterns of relationships among species. One envisioned that all species were descended from a single common ancestor, and that the history of life could be depicted as a branching tree, shown here. So this is a drawing from one of Darwin's notebooks written in 1837 as he developed his ideas on the origin of species. So we kind of envision a tree with twigs being those species on those twigs branching out as a tree as it grows. In Darwin's view, the twigs of the tree represent existing species. As one works down the tree, the joining twigs and branches reflects the pattern of common ancestry back in time to the single common ancestor of all life. The process of descent with modification from common ancestry results in all species being related in this branching, hierarchical fashion, and their evolutionary history can be depicted using branching diagrams referred to as phylogenetic trees. That is now shown here. So it shows how evolutionary relationships are depicted with a branching diagram. Humans and chimpanzees are descended from a common ancestor and are each other's closest living relative. Humans, chimps, and gorillas share older common ancestor at node 2. And all apes share a more distant common ancestor shown at node 3. One key to interpreting a phylogeny is to look at how recently a species share a common ancestor rather than looking at the arrangement of species across the top of the tree. If you compare the three versions of the phylogeny shown, you can see the relationships are the same. Regardless of where they are positioned, chimpanzees and humans are still more closely related to each other than to any other species. Moreover, even though humans are placed next to gibbons in version 1, the pattern of relationships still indicates that humans are more closely related with gorillas and orangutans than with gibbons. Phylogenies are also sometimes displayed on their side rather than upright, but this arrangement does not affect its interpretation. We might expect that the greater the time since two species diverge from a common ancestor, the more different they would be. Early systemicists relied on this reasoning and constructed phylogenies based on the overall similarity. If in fact species evolved at a constant rate, then the amount of divergence between two species would be a function of how long they had been diverging, and thus phylogenies based on degree of similarity would be accurate. As a result, we might think that chimps and gorillas are more closely related to each other than either is to humans. But as Chapter 22 revealed, evolution can occur very rapidly at times and very slowly at others. Species invading new habitats are likely to experience new selective pressures and may change greatly. Those staying in the same habitats as their ancestors may change only a little. For this reason, rates of change may vary markedly among species. Consequently, some will diverge more than others, and as a result, similarity is not necessarily a good predictor of how long it has been since two species shared a common ancestor. A second fundamental problem exists as well. Evolution is not always divergent. In Chapter 21, we discuss convergent evolution, in which two species independently evolve the same features. Often species evolve convergently because they use similar environments, in which similar adaptations are favored. As a result, two species that are not close related may end up more similar to each other than they are to their close relatives. Evolutionary reversal, the process in which a species re-evolves the characteristics of ancestral species, also has this effect. On to cladistics. Because phenotypic similarity may be misleading, most systemicists no longer construct their phylogenetic hypotheses solely on this basis. Rather, they distinguish similarity among species that is inherited from the most recent common ancestor of an entire group, which is called ancestral, from similarity that arose more recently, that is not inherited from the most recent common ancestor group, and is shared only by a subset of the species. This similarity is called derived. In this approach, termed cladistics, only shared derived characters are considered informative in determining evolutionary relationships. To employ this method of cladistics, systemicists first gather data on a number of characters for all the species in the analysis. Characters can be any aspect of the phenotype, including morphology, physiology, behavior, and DNA. As chapters 18 and 24 show, the revolution in genomics is now providing a vast body of data, revolutionizing our ability to identify and study character variation. Useful, the characters should exist in recognizable character states. For example, consider the character tail in vertebrates. This character has two states, present in most vertebrates and absence in humans, apes, and some other groups, such as frogs. A cladistic analysis begins by determining the states for a number of characters for each taxon. Taxa are species or higher level groups such as genera or families. So it says for each taxon in the analysis. In the example in figure 23.2, shown here now, 6 characters are used for 7 taxa. The first character, presence of jaws, is scored as absent in the lamprey, denoted by a 0, and present in other species, denoted by a 1. Another character, hare, is absent in all of the taxa except the 3 mammal species. The presence of hare is a shared derived feature of mammals. In contrast, the presence of lungs in mammals is an ancestral feature because it is also present in amphibians and reptiles, and therefore presumably evolved prior to the common ancestor of mammals. The presence of lungs therefore does not tell us that mammal species are more closely related to one another than to reptiles or amphibians, but the shared derived feature of hare suggests that all mammal species share a common ancestor that existed more recently than the common ancestor of mammals, amphibians, and reptiles. To return to the question concerning the relationships of humans, chimps, and gorillas, the number of morphological and DNA characters exist that are derived and shared by chimps and humans, but not gorillas or other great apes. These characters suggest that chimps and humans diverged from a common ancestor that existed more recently than the common ancestor of gorillas, chimps, and humans. So let's look now at figure 23.2b. There you see what we mentioned there. Once the data are assembled, the first step in a cladistic analysis is to polarize the characters. That is, to determine whether particular character states are ancestral or derived. To polarize the character, tail for example, systemicists must determine which state, presence or absence, was exhibited by the most recent common ancestor of this group. Usually, the fossils available do not include the most recent common ancestor, or at least we cannot be confident that they do. As a result, the method of outgroup comparison is used to assign character polarity. To use this method, a species or group of species that is closely related to but not a member of the group under study is designated as the outgroup. When the group under study exhibits multiple character states, and one of these states is exhibited by the outgroup, then that state is considered to be ancestral, and the other states are considered to be derived. However, outgroup species also evolve from their ancestors, so the outgroup species will not always exhibit the ancestral condition. For this reason, polarity assignments are most reliable when the same character state is exhibited by several different outgroups. In the preceding example, a tail is generally present in the nearest outgroups of vertebrates. Consequently, the presence of a tail in vertebrates is considered ancestral, and its absence in some species is considered derived. Construction of a cladogram. Once all characters have been polarized, given that 0 or that 1, systemicists use this information to construct a cladogram, which depicts a hypothesis of evolutionary relationships. A cladogram is the type of phylogenetic tree produced by cladistic analysis. Species that share a common ancestor, as indicated by possession of shared derived characters, are said to be a clade. Clades are thus evolutionary units and refer to a common ancestor and all of its descendants. A derived character shared by clade members is called a synapomorphy of that clade. Figure 23b illustrates that a simple cladogram is a nested set of clades, each characterized by its own synapomorphies. For example, amniotes are a clade for which the evolution of an amniotic membrane is a synapomorphy. Within that clade, mammals are a clade with hair as a synapomorphy, and so on. Ancestral states are called plesiomorphies, and shared ancestral states are called symplesiomorphies. In contrast to synapomorphies, symplesiomorphies are not informative about phylogenetic relationships. Symplesiomorphies. Let's return to the character state presence of tail, which is exhibited by lampreys, sharks, salamanders, lizards, and tigers. Does this mean that tigers are more closely related to and shared a more recent common ancestor with lizards and sharks than to apes and humans, their fellow mammals? The answer, of course, is no, because symplesiomorphies reflect character states inherited from a distant ancestor. They do not imply that a species exhibiting that state are closely related. In real-world cases, phylogenetic studies are rarely as simple as the examples we have shown so far. The reason is that in some cases, the same character has evolved independently in several species. These characters would be categorized as shared derived characters, but they would be false signals of a close evolutionary relationship. In addition, derived characters may sometimes be lost as species within a clade revolve to the ancestral state. Homoplasy refers to a shared derived character state that has not been inherited from a common ancestor exhibiting that character state. Homoplasy can result from convergent evolution or from evolutionary reversal. For example, adult frogs do not have a tail. Thus, absence of a tail is a synapomorphy that unites not only gorillas and humans, but also frogs. However, frogs are not closely related to gorillas and humans. They have neither an amniotic membrane nor hair, both of which are synapomorphies for clades that contain gorillas and humans. In cases such as this, when there are conflicts among the characters, systemicists rely on the principle of parsimony, which favors the hypothesis that requires the fewest assumptions. As a result, the phylogeny that requires the fewest evolutionary events is considered the best hypothesis of phylogenetic relationships. In the example just stated, therefore, grouping frogs with salamanders is favored because it requires only one instance of homoplasy, whereas a phylogeny in which frogs were most closely related to humans and gorillas will require two homoplastic evolutionary events: the loss of both amniotic membranes and the loss of hair in frogs. The examples presented so far all involve morphological characters, but systemicists increasingly use DNA sequence data to construct phylogenies because the large number of characters that can be obtained through sequencing. Cladistics analyzes sequence data in the same manner as any other type of data. Character states are polarized by reference to the sequence of an outgroup, and a cladogram is constructed that minimizes the amount of character evolution required, shown here. If characters evolve from one state to another at a slow rate compared with the frequency of speciation events, then the principle of parsimony works well in reconstructing evolutionary relationships. In this situation, the principle's underlying assumption that shared derived similarity is indicative of recent common ancestry is usually correct. In recent years, however, systemicists have realized that some characters evolve so rapidly that the principle of parsimony may be misleading. Of particular interest is the rate at which some parts of the genome evolve. As discussed in Chapter 18, some stretches of DNA do not appear to have any function. As a result, mutations that occur in these parts of the DNA are not eliminated by natural selection, and thus the rate of evolution of new character states can be quite high in these regions as a result of genetic drift. Moreover, because only 4 character states are possible for any nucleotide base, adenine, cytosine, guanine, and thymine, there is a high probability that 2 species will independently evolve the same derived character state at any particular base position. If such homoplasy dominates the character data set, then the assumptions of the principle of parsimony are violated. Similarity of species in this situation would more likely result from homoplasy than from inheritance from a common ancestor, and as a result, under these circumstances, phylogenies inferred using the cladistic method are likely to be inaccurate. 2 approaches. Because evolution can sometimes proceed rapidly, systemicists in recent years have been exploring other methods based on statistical approaches such as maximum likelihood to infer phylogenies. These methods start with an assumption about the rate at which characters evolve and then fit the data to these models to derive the phylogeny that best accords, i.e., is maximally likely with these assumptions. One advantage of use of these methods is that different assumptions of the rate of evolution can be used for different characters. If some DNA characters evolve more slowly than others, for example, because they are constrained by natural selection, then the methods can employ different models of evolution for the different characters. This approach is more effective than parsimony in dealing with homoplasy when rates of evolutionary changes are high. Secondly, the molecular clock. In general, cladograms, such as the one shown in figure 23.2, indicate only the order of evolutionary branching events. They do not contain information about the timing of these events. In some cases, however, branching events can be timed, either by reference to fossils or by making assumptions about the rate at which characters change. One widely used but controversial method is the molecular clock, which states that the rate of evolution of a molecule is constant through time. In this model, divergence in DNA can be used to calculate the times at which branching events have occurred. To make such estimates, the timing of one or more divergence events must be confidently estimated. For example, a fossil record may indicate that 2 clades diverge from a common ancestor at a particular time. Alternatively, the timing of separation of 2 clades may be estimated from geological events that likely led to their divergence, such as the rise of a mountain now separates the 2 clades. With this information, the amount of DNA divergence separating 2 clades can be divided by the length of time separating the 2 clades, which produces an estimate of the rate of DNA divergence per unit of time, usually per million years. Assuming a molecular clock, this rate can then be used to date the divergence events in a cladogram. Although the molecular clock appears to hold true in some cases, in many others the data indicate that rates of evolution have not been constant through time across all branches in an evolutionary tree. For this reason, evolutionary dates derived from molecular data must be treated cautiously. Recently, methods have been developed to date evolutionary events without assuming that molecular evolution has been clock-like. These methods hold great promise for providing more reliable estimates of evolutionary timing. Systematics and classification. Whereas systematics is the reconstruction and study of evolutionary relationships, classification refers to how we place species and higher groups, genus, family, class, order, and so forth into taxonomic hierarchy. Taxonomy is the science of classifying living things. By agreement among taxonomists throughout the world, the first word of the binomial name is the genus to which the organism belongs. This word is always capitalized. The second word refers to the particular species and is not capitalized. The 2 words together are called the species name or scientific name and are written in italics. For example, Homo sapiens. Once a genus has been used in the body of a text, it is often abbreviated in later uses. For example, the dinosaur Tyrannosaurus rex becomes T. rex. Also by agreement among taxonomists, no two plant, animal, or microbial species have the same scientific name. The scientific name of an organism is the same anywhere in the world and avoids confusion caused by common names that may differ from one place to the next. Oddly, the same scientific name can be used for species in different kingdoms, but such duplication is very rare. The taxonomic system is hierarchical, with taxa at lower level grouped into smaller number of taxa in the higher level. So I'll say again, the taxonomic system is hierarchical, with taxa at a lower level grouped into smaller number of taxa in the higher level. Just as species are grouped into genera, genera with similar characteristics are grouped into families, and similar families are placed into the same order. Orders with common properties are placed into the same class, and classes with similar characteristics are into the same phylum. Plural is phyla. And then finally, the phyla are assigned to one of several great groups called kingdoms. In addition, an eighth level of classification called a domain is now frequently used. If you check out what is called Figure 23.5, it illustrates the classification system using the gray squirrel as an example. So the gray squirrel is in domain Eukarya, kingdom Animalia, phylum Chordata, subphylum Vertebrata, class Mammalia, order Rodentia, family Sciuridae, genus Sciurus, and is called Sciurus caroliniensis. That is the gray squirrel, and that is some very small text. The categories at different levels may include many, a few, or only one taxon. For example, there is only one living genus of the family Ornithorhynchidae, containing the species O and Antennas, the duck-billed platypus, but several living genera of Fagaceae, the birch family. To someone familiar with classification or having access to the appropriate reference books, each taxon applies both a set of characteristics and a group of organisms belonging to the taxon. Systematics and the traditional classification are not always congruent. To understand why, we need to consider how species may be grouped based on their phylogenetic relationships. A monophyletic group includes the most recent common ancestor of the group and all of its descendants. By definition, a clade is a monophyletic group. A paraphyletic group includes the most recent common ancestor of the group and some, but not all, descendants. And a polyphyletic group does not include the most recent common ancestor of all members of the group. It merely includes descendants. Taxonomic hierarchies are based on shared traits, and ideally they should reflect evolutionary relationships. Traditional taxonomic groups, however, do not always fit well with the new understanding of phylogenetic relationships. For example, birds have historically been placed in the class Aves, and dinosaurs have been considered part of the class Reptilia. But recent phylogenetic advances make clear that birds evolved from dinosaurs. The last common ancestor of all birds and a dinosaur was a great meat-eating dinosaur. Therefore, having two separate monophyletic groups, one for birds and one for reptiles, including dinosaurs and crocodiles, as well as lizards, snakes, and turtles, is not possible. And yet terms Aves and Reptilia are so familiar and well established that suddenly referring to birds as a type of dinosaur and thus a type of reptile is difficult for some. Nonetheless, biologists increasingly refer to birds as a type of dinosaur and hence a type of reptile. Situations like this are not common. Another example concerns the classification of plants. Traditionally, three major groups were recognized: green algae, bryophytes, and vascular plants. However, recent research reveals that neither green algae nor the bryophytes constitutes a monophyletic group. Rather, some bryophyte groups are more closely related to vascular plants than they are to other bryophytes, and some green algae are more closely related to bryophytes and vascular plants than they are to other green algae. As a result, systemicists no longer recognize green algae or bryophytes as evolutionary groups, and the classification system has been changed to reflect evolutionary relationships. Chapter 22. You read about a number of different ideas concerning what determines whether two populations belong to the same species. The biological species concept defines species as groups of interbreeding populations that are reproductively isolated from other groups. In recent years, a phylogenetic perspective has emerged and has been applied to the question of species concepts. Advocates of the phylogenetic species concept propose that the term species should be applied to groups of populations that have been evolving independently of other groups of populations. Moreover, they suggest that phylogenetic analysis is the way to identify such species. In this view, a species is a population or a set of populations characterized by one or more shared derived characters. The rationale being that the possession of a derived character implies a period of separate evolution. This approach solves two problems with the biological species concept that were discussed in Chapter 22. First, the biological species concept cannot be applied to allopatric populations because scientists can't determine whether individuals of the populations would interbreed and produce fertile offspring if they ever came together. The phylogenetic species concept solves this problem instead by trying to predict what would happen in the future if allopatric populations ever came into contact. The phylogenetic species concept looks to the past to determine whether a population or groups of populations have evolved independently for a long enough time to develop its own derived characters. Second, the phylogenetic species concept can be applied equally well to both sexual and asexual species, in contrast to the biological species concept, which deals only with sexual forms. Phylogenetic species concept has problems. Critics contend that it will lead to the recognition of even slightly different populations as distinct species. And then, along that same note, it mentions a paraphylil problem, which I won't get into the paraphylil problem. One thing I should also mention along this would be the basis for all comparative biology, that are homologous structures. And this was already given, but a brief review, derived from the same ancestral source, such as the dolphin's flipper and the horse's leg, whereas homoplastic structures are not. In other words, the wings of birds and those wings of dragonflies. Those are not homologous. So let us begin. Phylogenies not only provide information about evolutionary relationships among species, but are also indispensable for understanding how evolution has occurred. By examining the distribution of traits among species in the context of their phylogenetic relationships, much can be learned about how and why evolution has proceeded. In this way, phylogenetics is the basis of all comparative biology, as I was mentioning. In Chapter 21, we point out that homologous structures are those that are derived from the same body part in a common ancestor. And I mentioned that the forelegs of a dolphin, its flipper, and of a horse, its leg, are homologous because they are derived from the same bones in an ancestral vertebrate. By contrast, the wings of birds and those of dragonflies are homoplastic structures because they are not derived from the same structure in a common ancestor. Phylogenetic analysis can help determine whether structures are homologous or homoplastic. Recent fossil discoveries have revealed that many species of dinosaurs exhibited parental care. They incubated eggs, laid in nests and took care of the growing baby dinosaurs, many of which could not have fended for themselves. Some recent fossils show dinosaurs sitting on a nest in exactly the same posture used by birds today. See figure 29. See figure 23.9a. Shown here to the left. So if you look here closely, you can see that the dinosaur is there. And the dinosaur is not only there, but there on those eggs shown to the right-hand side. Initially, these discoveries were treated as remarkable and unexpected. Dinosaurs apparently had independently evolved behaviors similar to those of modern-day organisms, but examination of the phylogenetic position of dinosaurs indicates that they are most closely related to two living groups of animals, the crocodiles and birds, both of which exhibit parental care. It appears likely, therefore, that the parental care exhibited by crocodiles, dinosaurs, and birds did not evolve convergently from different ancestors that did not exhibit parental care. Rather, the behaviors are homologous, inherited from each of these groups from their common ancestor that cared for its young. In other cases, by contrast, phylogenetic analysis can indicate that similar traits have evolved independently in different clades. This convergent evolution from different ancestral sources indicates that such traits represent homoplasies, and as a result, the fossil record reveals that extremely elongated canine teeth, saber teeth, occurred in a number of different groups of extinct carnivorous mammals. Although how these teeth were actually used is still debated, all saber-toothed carnivores had body proportions similar to those of cats, which suggests that these different types of carnivores all evolved into a similar predatory lifestyle. Examination of the saber-toothed character state in a phylogenetic context reveals that it most likely evolved independently at least 4 times. And this shows such in figure 23.10. Conducting tubes in plants provides a similar example. The tracheophytes, a large group of land plants discussed in chapter 29, transport photosynthetic products. And you see the 4 times the saber tooth has evolved. Back to where I was with plants. So the tracheophytes, a large group of land plants discussed in chapter 29, transport photosynthetic products, hormones, and other molecules over a long distance through elongated tubular cells that have perforated walls at the end. These structures are stacked upon each other to create a conduit called a sieve tube. Sieve tubes facilitate long-distance transport that is essential for the survival of all land plants, or the survival of tall plants on land. Most members of the brown algae, which includes kelp, also have seed tube elements that aid in the rapid transport of materials. The land plants and brown algae are distantly related, and their last common ancestor was a single-celled organism that could not have had a multicellular transport system. This indicates that the strong structural and functional similarity of the sieve tube elements in these plant groups is an example of convergent evolution. So one thing I will mention is that phylogenetics helps explain species diversification. One of the central goals of evolutionary biology is to explain patterns of species diversity. Why do some types of plants and animals exhibit more species richness, a greater number of species per clade than others? Phylogenetic analysis can be used both to suggest and to test about such differences. As earlier mentioned, beetles from order Coleoptera are the most diverse group of animals. Approximately 60% of all animal species are insects, and there are far more species of beetles than any other type of insect. Among beetles, families that are herbivorous are particularly species rich. Examination of the phylogeny provides insight into beetle evolution diversification, and figure 23.16 shows such a thing. Among the Phytophaga, the clade that contains the most herbivorous beetle species, the deepest branches belong to beetle families that specialize on conifers. This finding agrees with the fossil record because conifers were among the earliest seed plant groups to evolve. By contrast, the flowering plants, the angiosperms, evolved more recently in the Cretaceous, and beetle families specializing on them have shorter evolutionary branches, indicating their most recent evolutionary appearance. This correspondence between phylogenetic position and timing of plant origin suggests that beetles have been remarkably conservative in their diet. The family Pneumonicaceae for example, appears to have remained specialized on conifers since the beginning of the Jurassic, which approximately 210 million years ago. So you have conifers there shown in red, cycads shown in blue, which is also a form of gymnosperm, and then lastly are the angiosperms. So as mentioned moments ago, you can see just how deep the tree is, how deep the branches are in red. The phylogenetic perspective suggests factors that may be responsible for the incredible diversity of beetles. The phylogeny for the Phytophaga indicates that this is not the evolution of herbivory itself that is linked to great species richness. Rather, specialization on angiosperms seem to have been a prerequisite for great species diversification. Specialization on angiosperms appear to have arisen 5 times independently within herbivorous beetles. In each case, the angiosperm specializing clade is substantially more species-rich than the clade to which it is most closely related, termed sister clade, which specializes on some other type of plant. While specialization on angiosperms has led to great species diversity, it is not yet clear and is the focus of much current research. One possibility is that this diversity is linked to the great species richness of angiosperms themselves, with more than 300,000 species of angiosperms, and those are called flowering plants. Beetle clades specializing on them may have had a multitude of opportunities to adapt to feed on different species, thus promoting species divergence and speciation. To end the chapter, let's get to disease evolution. The examples so far have illustrated the use of phylogenetic analysis to examine relationships among species. Such analyses can also be conducted on virtually any group of biological entities as long as evolutionary divergence in these groups occurs by a branching process with little or no genetic exchange between different groups. No example illustrates this better than recent attempts to understand the evolution of viruses. In this section, we will first see how phylogenetic analysis has been important in discovering the origin and evolution of HIV, the virus that causes the acquired immunodeficiency syndrome, AIDS. Then we will see how the same approach was proven critical in understanding the recent coronavirus pandemic. AIDS was first recognized in the early 1980s, and it rapidly became epidemic in the human population. Current estimates are that more than 38 million people are infected with the human immunodeficiency virus, that's HIV, of whom nearly 700,000 die each year. At first, scientists were perplexed about where HIV had originated and how it infected humans. In the mid-1980s, however, scientists discovered a related virus in laboratory monkeys termed simian immunodeficiency virus, that's SIV. In biochemical terms, the viruses are very similar, although genetic differences exist. At last count, SIV has been detected in more than 40 species of primates, but only in species found in sub-Saharan Africa. Interestingly, SIV, which appears to be transmitted sexually, does not appear to cause illness in some of these species. Based on the degree of genetic differentiation among strains of SIV, scientists estimate that SIV may have been around for more than 1,000,000 years in these primates, perhaps providing enough time for these species to adapt to the virus and prevent it from having adverse effects. Phylogenetic analysis of strains of HIV and SIV reveals 3 clear findings. First, HIV obviously descended from SIV. All strains of HIV are phylogenetically nested within clades of SIV strains, indicating that HIV is derived from SIV. And it's shown here for you in figure 2317. And if you're wondering what's being shown here, I'll say again that HIV in those instances shown is nested within a clade with SIV in each instance there. Second, a number of different strains of HIV exist, and they appear to represent independent transfers from different primate species. Each of the human strains is more closely related to a strain of SIV than it is to other HIV strains, indicating separate origins of the HIV strains. Finally, humans have acquired HIV from different host species. HIV-1, which is the virus responsible for the global epidemic, has 4 subtypes. 2 of these subtypes are more closely related to strains in chimpanzees, while the third is more closely related to a strain in gorillas, indicating transmission from both ape species. The origin of the 4th subtype is not clear. Further sampling will likely reveal it to be a sister taxon to a currently undiscovered chimp or gorilla strain. By contrast, subtypes of HIV-2, which is much less widespread, in some cases known from only one individual, are related to SIV found in West African monkeys, primarily the sooty mangabe, and moreover, the subtypes of HIV-2 also appear to represent multiple cross-species transmissions to humans. Several hypotheses have been proposed to explain how SIV jumped from chimps to monkeys. The most likely idea is that transmission occurred as a result of blood-to-blood contact that may occur when humans kill and butcher monkeys and apes. Recent years have seen a huge increase in the rate at which primates are hunted for the bushmeat market, particularly in Central and Western Africa. This increase has resulted from growing human populations desiring greater amounts of protein combined with increased access to the habitats in which these primates live as a result of the road building and economic development. The unfortunate result is that population sizes of many primate species, including our closest relatives, are plummeting toward extinction. A second consequence of this hunting is that humans are increasingly brought into contact with bodily fluids of these animals, and it is easy to imagine how during butchering process, blood from a recently killed animal might enter the bloodstream through open sores or cuts in the skin. Where and when did this cross-species transmission occur? HIV strains are the most diverse in Africa, and the incidence of HIV is higher there than elsewhere in the world. Combined with the evidence that HIV is related to SIV in African primates, it seems certain that AIDS appeared in Africa first. As for when the jump from other primates to humans occurred, the fact that AIDS was not recognized until the 1980s suggests that HIV probably arose recently. Descendants of slaves brought to North America from West Africa in the 19th century lacked the disease, indicating that it probably did not occur at the time of the slave trade. Once the disease was recognized in the 1980s, scientists scoured repositories of old blood samples to see whether HIV could be detected in blood samples from the past. The earliest HIV positive result was found in a sample from 1959, pushing the date of origin back at least 2 decades. Based on the amount of genetic difference between strains of HIV-1, including the 1959 sample, and assuming the operation of a molecular clock, scientists estimate that the deadly strain of AIDS probably crossed into humans in the 1920s. The AIDS virus evolves extremely rapidly, so much so that different strains can exist within a single individual in a single population. As a result, phylogenetic analysis can be applied to answer very specific questions. Just like phylogeny proved useful in determining the source of HIV, it can also pinpoint the source of infection for particular individuals. This ability became apparent in a court case in Louisiana in 1998, in which a dentist was accused of injecting his former girlfriend with blood drawn from an HIV-infected patient. The dentist's records revealed that he had drawn blood from the patient and had done so in a suspicious manner. Scientists sequenced the viral strains from the victim, from the patient, and from a large number of HIV-infected people in the local community. The phylogenetic analysis clearly demonstrated that the victim's viral strain was most closely related to the patient's. And you can see this in the figure shown here, figure 2318. And this analysis, which for the first time established phylogenetics as a legally admissible form of evidence in the courts in the United States, helped convict a dentist who is now serving a 50-year sentence for attempted murder. And lastly, SARS-CoV-2, COVID-19, emerged in late 2019. And scientists had better tools for DNA sequencing than for early HIV. So it was quickly identified as a coronavirus. It was similar to SARS from the 2003 outbreak, which you all may not have been alive back then. So phylogenetic analysis showed most closely related viral lineages from a bat. So the disease apparently emerged in a market in Wuhan, China, with many live wild animals but no bats. So it's possibly an intermediate species got the virus from a bat and then passed it on to humans, like the civets with SARS in 2003. So to not take too much time to end this lecture, I'll say that when SARS-CoV-2, COVID-19 emerged in early 2020, scientists were well prepared to investigate its origin and spread following the approaches established to study HIV, which I just went over, and other emerging diseases over the previous several decades. Moreover, the capacity to collect DNA sequence data rapidly and in large quantities had greatly expanded since the emergence of earlier diseases. So, as mentioned, it was very quickly that the virus was responsible for the disease was identified as a coronavirus, similar to SARS from 2003. So, one complication was that the disease first emerged in a market in Wuhan, China, where live animals of many species were kept for sale in crowded conditions. So the phylogenetic analysis of genetically different strains of COVID-19 in different patients has been used to understand how the disease spread, just as with HIV. One study among patients in California early in the pandemic indicated that already by March of 2020, at least 7 different strains had arrived in the state, indicating the introduction of the disease from multiple localities on several continents. On the other hand, clusters of patients possessing the same strain indicated the occurrence of community transmission from one individual to another. A similar phylogenetic analysis of patients in New York City in March of 2020 identified at least 9 introductions, most from Europe or elsewhere in North America, and multiple cases of community transmission. So hopefully you've learned a lot about systematics, phylogenies, and comparative biology. Prepare well for your test class. If I can help you, let me know. And this has been your instructor, Skylar Huff. Thank you all for listening and learning.